Hello, everyone, and welcome. My name is Jennifer Nyman. I'm a principal at Geosyntech Consultants and the coordinator of the webinar series on behalf of SERDP and ESTCP. Today's webinar focuses on abiotic degradation of chlorinated solvents in the subsurface. First, Dr. Paul Trotniak will discuss some of the core concepts of abiotic natural attenuation by reactive minerals. Paul's presentation will be followed by a question and answer session. Next, Dr. Michelle Scherer will discuss abiotic natural attenuation of PCE and TCE by reactive minerals. Her presentation will also be followed by a question and answer session. We will conclude the webinar with a longer Q&A session with both of today's speakers. The next few slides provide some instructions on optimizing your webinar experience. If you haven't done so already, you can download Zoom at the link shown here. If you cannot download Zoom, you can view the slides using a compatible internet browser like Firefox or Edge. If you are unable to view the slides or if your screen freezes, you can try keying Control and F5 to perform a hard refresh. If you are accessing the audio through your computer, you can click on the arrow next to the Join Audio button to test the speaker and microphone to be able to troubleshoot your audio. If you have any further difficulties, you can call into the conference line shown here. You can also submit a comment using the chat box. Please use the chat box, though, for comments only related to technical difficulties. We're going to use the Q&A option for questions for the speakers. If you have continued technical difficulties, you can download a PDF of the slides from the webinar webpage and call into the conference line provided to you in your webinar registration confirmation email. We will also be live streaming the webinar on the SERDP and ESTCP YouTube channel at the link shown here. Today's broadcast will be listen only. You may submit questions by using the Q&A box on your screen. You do not need to wait until the Q&A periods to submit your questions. And in fact, we encourage you to submit questions in advance of that period. When submitting your questions, please make sure to add your organization name at the end of your question so that we can identify you during the sessions. And with that, I would like to introduce Dr. Andrea Leeson, who is the Deputy Director of SERDP and ESTCP, as well as the Program Manager for the Environmental Restoration Program area. Dr. Leeson has been with SERDP and ESTCP since 2001. Prior to that, she was a research leader at the Battelle Memorial Institute, where she conducted research on in situ bioremediation and the design and implementation of innovative biological, chemical, and physical treatment technologies for site remediation and industrial wastewater. Andrea received her doctoral degree in environmental engineering from the Johns Hopkins University. Andrea, please proceed. Andrea, do we have you on the line?
I'm sorry, I'm here. I was having some technical difficulties with my computer, but I am back. You can hear me all right now?
Yes, it's clear.
Great. Thank you, Jennifer, and thank you to everyone who joined the webinar today. What I'd like to do before we jump into our technical talks today is just give you a little bit of an overview about who we are in terms of SERDP and ESTCP, and then I'll turn you over to our speakers today. We are two programs, sister programs, SERDP and ESTCP. SERDP is our Strategic Environmental Research and Development Program. This is really our science and technology program. It is a partnership between DoD, Department of Energy, the Environmental Protection Agency. And what we're really looking to do there is we fund fundamental to applied research, really to impact our real world environmental management issues at our facilities. ESTCP is the companion program, which is the Environmental Security Technology Certification Program, and this is our demonstration and validation program. And what we do is we take the research and the knowledge that comes out of SERDP or other research programs, and once it's mature, we demonstrate that those technologies in the field or at some sort of more larger scale. And at the end of that, what we really want to do is transition that technology out of the lab to end users. We have two main environmental drivers that really, where our research is really focused. First is the issue of sustaining testing and training ranges, facilities, and operations. And this is very broad. It can take focus on everything from maritime sustainability to noise issues on installations, sustainable forward operating bases, as well as our unexploded ordnance and the munitions constituents associated with them. We also deal with the issue of reducing the current and future liability. We have a large program that deals with what you could consider more traditional pollution prevention, where we're trying to eliminate the hazardous materials or processes on our installations. And then we're also dealing with contamination from past practices. And our talks today are really going to be tying into that issue that deals with not only our groundwater, but also soil, sediments, and some of these emerging contaminants. We take the technology transition very seriously, and we have a number of ways in which we look at that. We have videos, in-person training, many different guidances and manuals, and then of course we have our webinar series that you're participating in today. Listed here are many of the upcoming webinar series, and you can just look at this short list. You can see that there's a tremendous variability in the sort of topics that we cover. Every webinar that we have is archived on our website, so you can go back to the website and listen to those webinars if you weren't able to participate when they were live. If you go to this link, then you can see both the archived webinars, and you can also register for the upcoming webinars that may be of interest to you. What I'd also like to mention then is our symposium that is coming up very shortly, immediately after Thanksgiving. In the chat room, you can see the link to get more information about the symposium. The symposium is a fantastic opportunity to see everything that we're funding under SERDP and ESTCP. All of our investigators, well, I will say nearly all, provide at a minimum a poster, and then we have a number of technical talks as well. This year we're all virtual, but the format is, other than being virtual, is pretty similar to past symposia, with a number of technical presentations in all the different areas that we are funding research, as well as posters associated with those different topics. So at this point, Jennifer, I'll turn it back to you to introduce our next speaker.
Wonderful. Thank you, Andrea. It is now my pleasure to introduce our first speaker, Dr. Paul Trotniak, who is a professor in the School of Public Health at the Oregon Health and Science University in Portland, Oregon, where his research focuses on zero-valent iron for the remediation of contaminated groundwater, as well as more broadly aspects of in situ chemical reduction and oxidation. Most of his recent and ongoing SERDP projects relate to the characterization of in situ redox processes involved in situ chemical reduction and abiotic natural attenuation. Prior to joining OHSU, he served as a National Research Council postdoctoral fellow at the United States Environmental Protection Agency Laboratory in Athens, Georgia, and he served as a research associate at the Swiss Federal Institute for Water Resources and Water Pollution Control, or EVOG. Dr. Trotniak has a doctoral degree in applied chemistry from the Colorado School of Mines. Paul, please proceed.
So thank you to Andrea and to Rula for inviting me to give this webinar today, together with Michelle. The two projects that will be presented today are nicely complementary, and so Michelle and I conferred a little bit about how to arrange our scope, and we came up with this. So the overarching theme is abiotic attenuation by reactive minerals, and the first part is mine, and we're calling that core concepts, which mirrors what was in my project ER 2620. And the second part we're calling case studies, which is along the lines of what was in Michelle's project ER 2532. My part's going to have two parts. The first one is going to be very general about abiotic natural attenuation and in situ chemical reduction, how they're defined, related, etc. And then the three core themes will be the part that's in green, which I'll elaborate momentarily. And then the second part of my presentation will focus on this idea of reactive mineral intermediates. So I want to start by going way back to put this in a broader historical context. The first point that I want to make is that the idea of abiotic reduction of contaminants is not even remotely new. This bottom of the slide here is a screenshot from the very first paper that I was involved in as a graduate student. So this was volume 1, issue 1, page 1 of the Journal of Contaminant Hydrology in 1986. And together with my thesis advisor, Don McAlety, we wrote this review titled Abiotic Reactions of Anthropogenic Organic Chemicals in Anaerobic Systems. And the paper covers a variety of different kinds of contaminants, including chlorinated solvents, and most of what's reviewed in that 28-page review is what we would now call abiotic natural attenuation. So you might wonder, well, given that idea has been around for so long, why is it only relatively recently that it's become something that the consulting and practitioner community has been well aware of? And there's a variety of reasons for that change. One of them I want to point out is the intersection between abiotic natural attenuation and what we're calling now in situ chemical reduction, and in particular the leading reductant in ISCR being zero valent iron. In the development and adoption and now widespread use of ZVI as a groundwater treatment technology, I think has made it more familiar to the practitioner community that these abiotic processes are a natural thing, and then people are much more comfortable with it. And then that's translated to a greater awareness of the ANA scenarios, which are not necessarily involving chemical added reductants. So this Venn diagram shows the intersection between the two conceptually, and this slide here illustrates the same thing, but more in terms of the technological jargon that consultants and practitioners are familiar with. So on the left side, I have some technologies that are primarily natural intrinsic reductant based. So these would be ANA. And on the right side, I have some technologies that are more along the lines of ISCR because they're based on stronger chemical reductants. And so on the left side, ANA is the sort of umbrella category. And then below that, we have biogeochemical reductive dechlorination and in situ biogeochemical transformation, to related ideas that basically are enhanced versions of abiotic natural attenuation. On the right side, we have a variety of different types of technologies that are based on adding chemical reductants. The top one, called in situ redox manipulation, employed dithionite. The second one, EHC and Deramin, are trademarked products that have been developed and are based on a combination of a little bit of ZVI and some carbon, and they're designed to generally or to create reducing conditions by amendment. And then at the bottom, we have the really strong reductants, straight up ZVI, or also catalytic reductive dechlorination, which is hydrogen plus palladium. So now, a while ago, I wrote a review paper about in situ chemical reduction, and we took the ideas that were in the previous slide and tried to classify them into a scheme that could be used to sort of put some perspective on how these technologies were related to one another. And we came up with this figure. This figure is in reference 1 on the bibliography slide at the end. That'll be slide number 38. So you can look it up when we get to that. The figure shows that on the x-axis, pretty much the same thing you saw in the previous slide, a continuum of reductant strength, with the mildest reductants being the natural ones on the left side, and the strongest reductants being the highly engineered chemical reductants on the right side. We also have on the y-axis another variable, which we called treatment volume. Maybe we should have called it treatment concentration or something, but the idea is that on the top, we have source zone treatment, and at the bottom, we have dilute plume treatment. And we spread out these technologies into this space, and this classification scheme turned out to be surprisingly robust. And it's been a widely reproduced figure in a variety of documents since then. Just to get you oriented towards how it works, look at the lower left corner. Obviously, abiotic natural attenuation is a natural reductant-based technology that's applicable to dilute plumes. And in the upper right corner, SZTI stands for source zone targeted injection with nano ZVI. And that's obviously a highly engineered scenario, and it's directed towards source zones, and the others are distributed accordingly in that space. So given all that list of technologies, there's a lot of variations, right? So it can be a little bit overwhelming to try to keep track of what principles one might use to decide between and optimize these technologies. And so that's where this idea of core concepts comes in. And so I've tried to map that out with this diagram, where I've listed a bunch of ANA/ISCR technologies on the left. It's roughly the same as what was on the previous figure, plus a few others. And on the right, in blue, I've listed what I'm calling core concepts. And the idea of core concepts is just that they're the big ideas that are cross-cutting to all these technologies. And this is the kind of thing that generally that my research is focused on. And so I've listed many of them there. We don't have time to talk about most of them. Just to help illustrate what I mean by this, I've circled one, the one that's called intermediates and products from COC degradation. And the point there is that in the years since in situ chemical reduction and ANA have become widely familiar to the community, the idea that those pathways produce a sort of an abiotic signature in dechlorination that largely avoids the stall intermediates of cis, DCE, and vinyl chloride, and that's it. And as in contrast to the biologically mediated dechlorination pathways, that idea has become a huge paradigm shift in the field, and it's a big deal. It's a core concept, and so that's why it's listed there as such. However, I'm going to talk about 3 other core concepts, because those are the ones that were the focus of the project ER 2620. So there are 3 of them are listed here. The first one is what we call standardized metrics of remediation performance. The second one is enhancement and/or activation of remediation performance by mediation or activation. And the third one is the idea of reductant demand, as sort of the analogy to oxidant demand in ISCO. I just note that I have always decorated this slide with this picture from this textbook. The only reason it's there is to reinforce the idea that core concepts is a big idea, and that people write whole series of textbooks that have that in the title because that's appealing in the sense of having broad impact. Okay, so the first one, performance metrics. So the focus here is on the rate of contaminant reduction. And so we set out to do a number of things for the project. The first thing we did was compile a ton of data from what was previously available. And eventually, I'm going to make that openly accessible in an online database, but we haven't quite released that yet. We also did a bunch of experiments to fill some data gaps that we identified from the database compilation process. And so we've reported some of those data already. But the bulk of what was really important in this objective for the project was to figure out how to normalize those rate constants to make them more generally useful. And we started with the 2 variants that are highlighted in red, mass normalized and surface area normalized rate constants. You probably have all heard of this. This has been around for a long time now. I think of it as the standard kinetic model. And it's illustrated over on the right side with this figure where we plot log surface area normalized rate constant on the Y versus log mass normalized rate constant on the X. In this format, the diagonal lines are the specific surface area of the mineral. And this particular figure is for TCE, and it's color-coded according to other kinds of secondary variables like micron size ZVI versus nano ZVI, in impure ZVI versus pure ZVI, and various iron sulfide and iron oxide minerals without ZVI. There's a lot of details in the figure that we don't have time to go through here. This was published in a paper that we published as part of this project, which is reference number 4 in that bibliography slide that's coming up. And in there, we explain all the details of how we would interpret those. That paper, we were emphasizing somewhat the theme of the effect of sulfidation of these chemical reductants. And the things that are highlighted there in orange, that has to do with the discussion of the benefits of sulfidation. We also considered some other things. So we explored the possibility of magnetic susceptibility as being a tool for normalizing the kinetics of contaminant reduction. And we were not able to get that to work very well. And just to illustrate an example of the results that we got, this figure here shows a mass normalized rate constants for reduction of a variety of nitro compounds, so munitions related compounds, on the y-axis, and on the x-axis is mass normalized magnetic susceptibility. And this is routinely measurable, or easily measurable on even a field portable device, but unfortunately, there's really no apparent relationship between them. There's considerable variability in the rate constants, and that has due to things like pH and iron 2 concentration and other things. But there's no evidence for correlation between Km and magnetic susceptibility, and so you can't do a regression through that figure and come up with a magnetic susceptibility normalized rate constant. It's not going to work. So we also were interested in the idea of redox potential as a normalizing factor, and this has been more successful sporadically in the literature over the years. And so on the left side, there's a figure from a paper by Stewart and Gorski that is a dramatic illustration of how well it can work. However, keep in mind exactly what this is. This is surface area normalized rate constant for one model contaminant, nitrobenzene, and it comes from multiple sources. It includes different mineral oxides, and it's all, the x-axis is redox potential combined with pH, all determined for controlled batch reactors where it's basically pure minerals in otherwise deionized water. So that's a very controlled set of conditions to do these experiments. And the outliers that are down here in the lower right corner are actually where there was a bunch of carbonate in the system. And so as you can imagine, it's starting to deviate when the system gets more complex. We set out to try to explore whether we could make that work on environmental samples as part of a companion project with Rick Johnson. And that's shown on the figure on the right side. So this is mass normalized rate constant measured on samples from DoD sites that were sent to our lab, and we slurried them up and did batch experiments with the field sample. And we're plotting that versus redox potential determined on those samples with the help of an electron shuttle or mediator. And the different colored markers on that graph are different field sites and different treatment scenarios and stuff. And it's pretty clear that there's a promising relationship there, but that's still something that we're exploring. We haven't quite got to the point that this is a routinely applicable metric for scaling and field sites, but that may be on the horizon not so far from now. Okay, changing to the second core concept, activation and mediation. So we set out to explore the whole range of possibilities, and so we considered things like heat and light, weak magnetic field, but those things are only really very applicable under lab conditions. Electron shuttles of various sorts have been proposed over the years, and some of them are more field applicable than others. And we thought a lot about these and did a few experiments with them, including riboflavin, for example. And in the end, we concluded that we should focus on ligand stabilized iron 2 because that seemed to be producing the most dramatic effect. And you can see that on the figure on the right, where I've plotted rate constant with a treatment, an enhancement, an activation treatment on the y-axis versus the corresponding rate constant without that treatment on the x-axis. So in this particular format, the diagonal line is the ratio of with treatment to without treatment. So above the diagonal line R equals 1, that's an enhancement, and below the line R equals 1 is an inhibition of performance. And as you can see from the figure, that a lot of these treatments, the legend says light is the red dots and weak magnetic field is the orange dots, et cetera. They all produce some degree of enhancement, but the most dramatic result was that cluster of purple points in the upper right, and that's iron 2 that's ligand stabilized. And so we decided to focus on that, and then that evolved into this concept that we're calling reactive mineral intermediates, RMIs. And so the last part of this talk, I'm going to focus on that. The evidence for the significance of the reactive intermediates in my lab turned up in this paper that's shown on the left, which is reference 3 coming up. And in that paper, we were studying the formation of hydrogen from ZVI. And the key point in this figure is just that you see a bimodal kinetics. And the steep part at the beginning is due to a transient mineral phase that is used up as the experiment proceeds. On the right-hand side is TCE degradation in experiments that Michelle Shearer performed. And she'll talk about those in the next presentation, but I include it here because we saw her work at this time, right about the same time we were writing the paper on the left. And so the two things, and her work was infecting our thinking about this idea of reactive mineral intermediates. But the most dramatic illustration of this for those of you that are on the line and field oriented is this from a column experiment that Rick Johnson did also right about the same time. And here what you see is on the bottom blue, you see the rate of degradation of TCE in this column when the column had just recently been set up and minimally equilibrated. And the column consists of 1% magnetite and sand. And so magnetite alone, without extensive equilibration, wasn't degrading TCE to any significant degree. In the top, in the green dashed line, you see the rate constant for degradation of TCE in the column after it had been equilibrated for, I think, a year, almost 2 years. It had been running for a long time. It had developed all that black precipitate, we're going to call it now RMI, at the bottom part of that column. And that was producing quite rapid degradation of TCE with calculated half-lives on the order of days. When Rick turned the column off and then continued to measure the rate of TCE degradation, you see the red line, and it decays over time. So the column became static. The conditions that were necessary to create and sustain the reactive mineral intermediate were interrupted. And so the reactivity went down. So this really kind of illustrates nicely the idea that this reactive mineral intermediate phase is a metastable and transient phase, and you really need to maintain the conditions that made it in order to keep it going. So we have done some additional work of our own on this, and in my lab, in batch experiments, we decided, since other people were doing TCE, we would try to see whether we could see evidence for the RMI effect with nitroaromatics. And so we used nitrobenzene, but also we have data on DNAN and TNT, which I don't have in this presentation because the data set's not quite as complete. This figure is for nitrobenzene, and the green dots decreasing show reduction of nitrobenzene, and the green dots increasing from the bottom of that figure show the formation of products. So we have good mass balance, that's the asterisks along the top. But the key thing to see here is that with each incremental addition of iron 2, which are the dashed lines the 2 dashed lines that are in the middle of the figure, each time we spike it with iron 2, the rate of reduction of the nitroaromatic compound increases incrementally and unmistakably. And if you visually look at the bottles that are shown over on the right, you can see that there was clearly a precipitate that was forming during that time. We attribute that to what we're calling our reactive mineral intermediate. Now, of course, what I'd like to do is link the first core concept with the second core concept. And so we would like to have a metric for describing the effect of the reactive mineral intermediate. And that's really comes back to this idea of, can we normalize these race constants to something along the lines of redox potential? Because you can measure the redox potential on the system where the RMI has formed. And so we hope to get this to work. The figure that's shown on the right is data that's from a paper that we're writing up right at this time. And so once we get that paper written up, we will use that data to try to normalize the data on this previous slide. And if all that works, we'll have a really firm basis for predicting rates of RMI enhanced contaminant reduction under dynamic conditions. So the last core concept is the idea of reductant demand, and this one is one that we developed in great detail in a paper that's published in ESPI. This one is reference 5 in the slide that's going to come up in just a second. Since I'm a little over time, I won't elaborate on this one, but the paper describes the math for this thoroughly. So just to quickly summarize, ANA and ISCR overlap, share core concepts, and for the performance metric core concept could include mass and surface area normalized rate constants. That certainly works, but not so applicable in the field. Magnetic susceptibility normalized rate constants, probably not. ORP, normalized rate constants, maybe. That remains to be seen exactly. In terms of mediation and activation, certainly manipulating the system to influence the amount of iron 2 is certainly an attractive scenario and looks promising. And on the demand capacity front, we can certainly influence and improve the efficiency, electron efficiency of the process by sulfidation. I didn't elaborate on that, but that's in the review paper I showed. So benefits to DoD, I'll just emphasize only the last one on the lower left, which is that this core concepts theme is very broad. It's meant to be applicable as widely as possible so that all of these core concepts are usable for all variations of ANA and ISCR, and actually quite a lot of other remediation technologies also. And so I'll end with this slide here, which is that bibliography slide that I promised with the references that I mentioned. And so I'll take whatever questions I'm allowed to at this point.
Fantastic. Thank you, Paul. We have received a number of questions from the audience. I'd like to encourage the audience to continue to submit questions. Please use the Q&A box at the bottom of the screen. Our first question is about the column that you talked about that was incubated for more than 1 year. There are 2 questions related to that. What was the black precipitate shown in the photo? And was the column operated under sterile conditions, or were there microbes present?
So the first part is relatively easy. That column was one that received sulfate. There's microbial sulfate reduction going on to produce sulfide, and we presume the precipitates are predominantly iron sulfide. It could also be some mixture of magnetite in there too. And we haven't done a detailed mineralogical characterization of that material as of now. Of course, you can't do that while the experiment's running because it's sacrificial. So we may have more information on that later, but that's all we know for now. The other question was, what was the other part of the question? The mineral. Was it sterile? Or could there have been microbes present? Obviously, it wasn't sterilized, and so the sulfate reduction is microbially mediated. So in that sense, it's a classic example of in situ microbiology driving the biogeochemistry. Where the question is a little bit more ambiguous is, okay, to what extent might the biology that's going on in the column have contributed to the contaminant reduction behavior that we showed in the figure? And we think that it's not. We think that primarily what we're seeing is the ANA effect, but that's a hard, that's a slippery slope to try to we can't autoclave the column or anything. So and all these RMI phases that we're alluding to are going to be very labile and vulnerable to any sort of inhibitory treatment you might do. So that's a little tricky one. That's all I can say about it for now.
Okay, thank you. Have you looked at how much of the reactive mineral intermediate you might need to be able to achieve effective degradation of something like TCE? And how would you sustain that in the field?
So to try to maybe break that into 2 things. So on the idea of how much do you need, that fits really well in my third core concept. So reductant demand, right? So how much of the RMI do you need to do the job? But we didn't quite answer that question directly because the frame of reference shifted when we became convinced that the RMI is a dynamic metastable phase. So it's forming continuously and being used up continuously. So it's not a stable phase in the sense that you put so many grams of magnetite in the column, or you injected so many tons of ZVI into the subsurface. It's not a stable phase like that. It has to be formed in situ, and so that makes the question much harder to answer. If what I think the bottom line is that you need to sustain the conditions that favor the existence of the RMI. And if you do it, then the RMI will be there forever and you'll have all the capacity you need. But that's kind of like the next generation of thinking on this subject.
And related to that, you showed the graph of the decay or the decrease in the rate constant over time for TCE degradation in the column study. Why was there that decrease in the rate?
Well, that comes back to the same point that I was just making, is that we think of this reactive mineral intermediate as a steady state phenomena, right? And so you have to be making it, and it's made and it's consumed simultaneously. So anything you do to shut off the formation part of it, will cause it to be depleted. And all we know at this point is that turning off the flow stopped feeding whatever exactly what the ingredients were that resulted in sustained reactivity of the RMI. And we're still working on trying to clarify exactly what that might be. And certainly that's going to have huge implications for translating this into field practice. But the idea that this RMI is a metastable phase and it needs to be treated as such is actually brand new. I mean, it wasn't around just a few years ago.
Mm-hmm. Right. There's a question about the ferrous iron ligand complexes. What type of ligands were used?
That figure that I showed with the purple dots that you're remembering was actually work from Tim Strathman's papers. So he was using Tiron, T-I-R-O-N. It's a reagent grade ligand that's complexes well with iron. It's a very convenient and well characterized ligand for studying iron 2 complexes in lab experiments. I don't think anybody has ever suggested that ligand per se would be usable in the field. There are many studies that have explored various other kinds of ligands for stabilizing iron 2. Not just actually for ISCO, that's kind of a newish idea, but there's variations on that have existed in ISCO forever because people have been trying to stabilize iron 2 for Fenton-like processes, right? And that particular bit hasn't quite crossed over into ISCR yet, but I imagine it will. So if you're what the question is really looking for a recipe that's ready to be taken to the field, I don't quite know what the answer to that is, but there's plenty of possibilities.
Great, thank you. We've had a couple of questions about the data you showed on slide 28. You showed two graphs of rate versus redox potential, and they had opposite slopes. Why were their slopes opposite?
That's just the scaling of the x-axis. So there's actually nothing particularly interesting going on there. So one of them's got a negative in front of the E and the other one doesn't. So that's an easy
okay. Gotcha. Thank you. And then the next question is that relates to your description of a variety of technologies for enhancing ANA. Have you looked at whether there are limits, whether there's some sort of, for example, thermodynamic limit about as to how much those approaches can be improved?
I like that question. I've given another talk at I think a REMTech meeting in the past on the theme of, I think I called it extreme reductance. And I was exploring the idea of what the thermodynamic limits are to reduction. And I was playing up the analogy to oxidation. So, you know, how, if you look at a redox ladder for treatment technologies, there are some oxidants that appear to have extremely high oxidation potentials, and the reductant side of the continuum is not quite so well populated. There's plenty to be done there. As some of you might know, I've done a little bit of work with zero valent zinc. That's quite a bit stronger than any other reductant we're talking about today. There's even others like magnesium, which is even stronger. It's a fun topic, but I can elaborate on it in some future future talk, but can't go further with that here.
Okay, understood. Thank you very much, Paul, for the very interesting presentation and answering the questions. And to our audience, we will have Paul back at the end of today's webinar to answer additional questions. If you have any more for him, please specify that when you submit the question. At this time, I would like to introduce our second speaker, Dr. Michelle Scherer, who is the Donald E. Bentley Professor of Engineering in the Department of Civil and Environmental Engineering at the University of Iowa. Her expertise is in environmental geochemistry, and that includes redox reactions at the mineral-water interfaces and contaminant fate in soils and sediments. Her research on the abiotic attenuation of chlorinated ethenes was recently recognized with the 2018 SERDP Project of the Year award. She has served as a member of EPA's Environmental Engineering Scientific Advisory Board and as an associate editor of Environmental Science and Technology. She has a doctoral degree in environmental science and engineering from the Oregon Graduate Institute of Science and Technology. Michelle, please proceed.
Excellent. Thank you very much, Jennifer. And thank you, Rula and Andrea, for inviting me to do this webinar, particularly with Paul. I think several of you know, but for those of you who don't, I did get my PhD back in Oregon with Paul. So this is really fun to be giving a webinar with him 20 years after my PhD. So Paul set this up very nicely. So I'll need to go to slide 42. So Paul set this up nicely because he made it so that you could see the big picture of what we are going to talk about today. What I'd like to do is dive a little bit deeper into where did this notion of an RMI originate? So what are the observations that we had that sort of led us to propose this concept of being able to, instead of having a mineral in a sediment or an aquifer that was responsible for it, that you actually had to have some formation or precipitation of these intermediate metastable transient phases. And so the two case studies I'll talk about are where we looked at magnetite and we looked at structural iron 2 in clay minerals. And this was a collaboration with Anka Newman at Newcastle University. Okay, so I will start with the clay minerals, and I'll need to go to slide 43. So both magnetite and structural iron 2 and clay minerals have been implicated in being able to reduce chlorinated ethenes. There's been a lot of discussion in the literature, and I think among practitioners, that if you find magnetite in a sediment, that it would be evidence that you could use as a secondary line of evidence to support ANA at that site. What I'd like to present today and make the argument today that the presence of these is unlikely to be sufficient to support, to serve as a secondary line of evidence for ANA. And then I would like to make this, show you the evidence that active precipitation. Slide 44, please. So is ANA likely? So we suggest, so the take-home message, if you take nothing else from my talk today, the take-home message is, rather than looking for, the silver bullet of a specific mineral in an aquifer, I think it's going to be these conditions, these sustaining conditions that Paul kept mentioning, that creates the potential and the geochemical environment where precipitation of these minerals can occur. My working hypothesis for, I think, my group and for several groups right now is that that is going to be more predictive. And there's lots of pieces of evidence coming out, and so that's what we're going to try to explore here today. And as Paul said, we don't have a recipe yet, but we're working on it. And so, next slide, please. So the evidence for ANA by iron minerals. So let's take a step back and look at what's out there. So everybody listening to this webinar knows that the primary evidence is the plume footprint, right? So if you see a plume that you've seen transformation and you can't attribute it to biological, you don't see products forming, right? That's sort of your first, hey, what's going on here? The other sort of secondary lines of evidence that have been discussed is if we have the presence of these iron minerals. So you take a sample, you do XRD on it, you extract it, you look for some of these iron minerals and if they're present there. And then in the lab, there are some iron minerals that can reduce. You know, we see rapid reduction by things like iron sulfides and green rusts of these chlorinated ethines. Next slide. Okay, so I consider this the trifecta of evidence for ANA at the Twin Cities Army Ammunition Plant, and I think many of you are familiar with this work of Mark Ferry, Wilkenford, and Wilson, where, you know, they had a plume and there was lots of transformation going on, and when they modeled it with just advection and dispersion and didn't include degradation, they really couldn't capture the plume. But when they put in a in situ degradation coefficient of 0.6 years to the -1, they could capture the plume. And then they did microcosm studies where they autoclaved and didn't autoclave, and they took samples from the sediment and they found magnetite. And so then they said that this could be due to magnetite. And so it's certainly a reasonable line of thinking. But when you go and you look at some of the data that's available in the laboratory, I started to get concerned that possibly, you know, there was something else going on there. Next slide, please. So when we look at these reactions in the laboratory with magnetite, so if you look at the data that's out there, so this is not my data from my group, this is sort of the literature that's often cited to support this. So the Lee and Batchelor paper, some work by Liz Butler. But when you look closely at that data, the data is really either for cis-DCE or the PCE and TCE data. There's very, very little degradation, almost none, right? So it's like 3 to 4% they might see, to the point where they're, you know, they won't even quantify Ks, or they, if you look over here on this one, this is magnetite, you know, this is the products they're observing, and they're observing almost no products, right? This is a blow up of this here. Next slide, please. So what did we do? So we said, okay, I really want to look at this closer. Let's put some, you know, we do a lot of experiments like this in our lab. So we reacted magnetite with PCE and TCE in the laboratory to check whether they would be reactive. We can make magnetites as a range of stoichiometries, meaning the iron 2 contact can range. So I think when we think about magnetite, we have to think about it as a continuum of maghemite, which has no iron 2, but is the same structure, the same spinel structure as magnetite. And then magnetite, which has a significant amount of iron 2. You know, one is an iron 3 and has no, you know, reductive capability for things. And then one is got a lot of iron 2 that can reduce things quite fast. Next slide, please. And so what we found when we put magnetite in a bottle with PCE and TCE, and these are laboratory experiments, various stoichiometries, anoxic in a glove box. It's quite small magnetite. We varied the pH, we varied the iron, the magnetite loading. We see no reduction. And this is over 5 months, so this is a long time. We see no production of, you know, products, no ethene, ethane, no acetylene. And we have complete mass balance. So it's not like we're losing. You know, these are difficult experiments to do and not lose stuff, but we see nothing. No degradation products forming and no loss of PCE. So alone, we see no reduction. Next slide, please. However, if we add iron 2 to the bottles, we do start to see some reduction sometimes. And so these bottles on the left, so the one to the left is magnetite, and the one to the right is magnetite with a high concentration of iron 2. And so this is what in our lab we call logoscopy, right? It's a pretty powerful little technique. You can see visually something is happening in that bottle. And what we're seeing there is we're seeing the precipitation of ferrous hydroxide. It's often commonly referred to as white rust, and you see it there precipitating as a white phase. And so the student, John Culpepper, who did this work, came into my office and said, yeah, I think I'm only seeing it when I see this white stuff form. And so that comment is what has led us down this road. Next slide, please. Okay, so this is, I would say, the essence of where we lean our RMI idea on. And so I want to take a little bit of time and walk you through these figures. So the figure on the left is just K observed, rate coefficient versus saturation index. So this is, we just take, you know, the Fe2 plus concentration, we take the pH, right? We calculate the activity product, right? OH minus times Fe2 plus squared, and then we divide it by the solubility product. It's sort of a classic way of being able to tell if something's going to precipitate. And if it's greater than 0, right? So here's our SI at 0. And if it's greater than 0, theoretically, you thermodynamically will precipitate FeOH2 solid. And if you look at all of these, right? So up when we're below solubility, this is a K observed, right? Very little reactivity, and then boom, you hit 0, and then you start to measure reduction of it. And so that's one way to plot it, is to plot it as SI, and we also plotted it as a solubility diagram. So this is Fe2 versus pH. So this gray area here is where FeOH2 solid would precipitate. The white area is where FeOH2 solid wouldn't precipitate. And those red, so then we plotted. You know, we ran a bunch of experiments at different pHs, at different iron 2 plus concentrations, and we plotted it on here. And the circle, the red circle, is proportional to the rate coefficient, to how fast. So the big red circles, it went away the fastest. The small red circles, it went away the slowest. And you can see here we have, you know, a cluster of sort of fairly big circles here right on this line of solubility, you know, right where you're getting active precipitation. So I do want to point out, as my colleague and friend Rick Johnson always points out to me, grounds me back into the real truth of field work. So this is Fe2 concentration in molar, right? So here we have 10 to the negative 3 molar, so 1 millimolar. And so you're talking 2 milligrams per liter of iron. So, you know, a reasonable amount of iron. You know, up here, you're talking even higher. You know, these are at 20 milligrams per liter of iron 2 plus. So these are fairly high concentrations of aqueous iron 2 concentration. And then notice you don't really start to see, you know, we in our lab, we often refer to pH 8 as the magic pH because, you know, boy, right around pH 8 is where, you know, things start to precipitate. So the take-home message from this is that if we see it visually using our fancy leucoscopy technique, we see some reduction. If we look at the, you know, thermodynamics of it, okay, it fits with that. Only when we predict it will precipitate, does it precipitate? Always good to know thermodynamics works. And then when it does precipitate, we do see some reactivity. And then, of course, we like to go beyond leucoscopy, and we have also confirmed ferrous hydroxide with XRD as well as other techniques. Next slide, please. Okay, so these are all in bottles, and you know, this is the kind of work that we do primarily, but we of course have to take this and relate it to the field, right? Our goal is to try to help people figure out how they can better clean up sites or predict how a site will behave. So we can take the information we have, we can take how much magnetite and how much iron 2 we have in a bottle, we can assume some typical aquifer properties, we can assume the magnetite in the sediments, and from here we took a range that was typical of the Twin Cities ranges that they had seen, and we can go ahead and just, you know, this is just doing some multiplication and division, and we can figure out, we can estimate some rate coefficients, extrapolate these under the field conditions, and we can see those on the next slide. So for magnetite plus iron 2, we can estimate aquifer first order decay coefficients of about 0.1 to 4.2 inverse years, translating to half-lives of 2 months to 6 years. And so when I show those numbers, people are like, oh, well, those are reasonable, right? Those could do something. Those could be real and participate and contribute to ANA in real field settings. But I want to emphasize that this is with the addition of aqueous iron 2, right? So this is not magnetite alone. Magnetite alone, recall, we saw no reactivity. Next slide, please. Okay, so to sum up magnetite, and then we'll talk about some clay minerals. This is a paper we published, and it got the cover. And I really like to point out, because I think it's fun, that the first author on this paper, John Culpepper, is an artist, and he actually painted that cover art there that you see to the left. The take-home message from this is then when we have magnetite alone, we see no reduction. When we add iron 2, if we add it under pH and aqueous iron 2 conditions where ferrous hydroxide precipitates, then we see some reduction, and we can quantify the rates of those. Next slide. So now we'll go on to structural iron 2 and clay minerals, and this work was led by Anke Newman at Newcastle with her student Jim Entwistle. And so the story is quite similar. So I'm going to go through this a little bit quicker than I went through the magnetite story. But essentially, we saw what was happening with the magnetite, and you know, iron 2 in structural iron 2 in clay minerals is often invoked also as possibly contributing to ANA. So we wanted to go ahead and actually test that. We wanted to test, just like we tested the iron 2 in magnetite, the iron 2 in these structural clays. And Anke picked 2 clays. She picked a clay that was a high iron containing clay, a notronite, and then she picked a montmorillonite that was a low iron 2. She reduced them. You know, she's the clay expert, so she did all her very careful, because clays are annoying to work with, you know, reduction of these with dithionite. And then, so they have fully reduced, the iron in the clays are fully reduced to iron 2. And then her and Jim reacted them for 6 months. Again, nice mass balance, right? She's not losing anything. They're not losing anything from the bottle, and no indication of products. Next slide, please. Okay, so then Anke did the same thing we did. She started adding aqueous iron 2 to explore, okay, well, if I add aqueous iron 2, can I then observe some reduction? And indeed, they do see some reduction. And they have a fairly similar story to what we have. It's different in that they use 2 different types of clays, and they observe different behavior with those, which has some interesting implications. But basically what you see here is you see this distinct color change, right? So the bottles that are shown here, the first one is just the really low iron 2 concentration added. B and C are higher and higher iron 2 concentrations. And only when you get to C, where you have really high iron 2 concentrations, do you see a formation of an RMI. And to the right, we just track the C2 products that were formed. And the reduced clay, no RMI was formed. And then in the inset, we show the Mossbauer spectra. And so on the one above it, where we do believe we have an RMI formed, so that's what it looks like in Mossbauer, which, so anybody would look at that and be like, oh, whatever, that's a mess. But even Mossbauer people look at that and say, okay, that's a mess. So we don't know exactly what it is. You know, we do know that Anke does in some cases see some ferrous hydroxide also, like we do. Next slide. So the take-home message from their work is again this notion that when you have the structural iron 2 just in the clay minerals reduced, so she makes sure it's fully reduced to iron 2, there's no indication that any reduction of PCE or TCE occur. However, when they add iron 2, and they have to add enough iron 2, and they have to have, interestingly, the low iron clay mineral tends to need less iron 2 added because of an electron transfer process that doesn't occur. They need the precipitate itself that forms to have a fairly high iron 2 content. And once that has a high enough iron 2 content, they do indeed see a reduction of PCE and TCE. Next slide. Okay, so to kind of bring it full circle here, I think it is really important that we start moving our conversation. We need to move the conversation from finding the magic, you know, specific mineral in the aquifer that we're going to say, if X is there, then I can say I have secondary line of evidence for abiotic natural attenuation. I think it's a much more complex story. I think these primary minerals that are there are not very reactive or reactive at all. I think we need to have these conditions where we're actively precipitating things. So just looking for them in the solid materials, I think, is not sufficient. Next slide. And again, this is right there, those photos are why we think that we have to create conditions where things are precipitating. I think the visual photo, we can back a lot of this up with other fancy techniques, but that tells a lot of it right there. So you have to have the right condition to have these things form in the presence of these other iron minerals. That's facilitating it for sure, but the mineral itself is not responsible. Next slide, please. Okay, so if we can't just go look for it, right? What are we gonna do, right? How are we going to be able to go to a site and have any sense, right? That's the goal of whether ANA might be supported at that. Well, Paul touched on some of this data, and I've already talked through some of it. The one in the upper left-hand corner is by Stewart and Gorski, and again, it is very specific conditions. It's nitrobenzene, but this is remarkable that you can predict decades of research in a laboratory with multiple iron oxides present, synthesized by different people under different conditions, and that with this expression, which they derived, right, to fit nitrobenzene reduction, basically from the Nernst equation, but to be able to predict it off of just the EH and pH, I think that's a remarkable finding that, as Paul said, is pointing us in a new direction. And then also Paul's work, where Chris and Rick, where they looked at these, getting the redox potentials in the presence of electron shuttles and being able to predict. Both of those are fairly remarkable findings that I agree with Paul, like 5 years ago, we were not talking like this. And then this notion of the saturation index, right? This idea that, if you're above it and you have these conditions, then you can sustain reduction. Next slide, please. All right, so we full circle back to what was my goal of the talk today? And my goal was to really try to start moving the conversation in the direction of, instead of looking for specific minerals, we should be looking for aquifer and groundwater properties. We think these may be more predictive. We don't have a specific recipe to share yet, but we're working on that, so stay tuned. And then my last slide. So these are the conclusions. I think I've summarized them already. Magnetite is unlikely to reduce PCE and TCE. Therefore, the presence of it is not sufficient. I'll come out pretty strong that I think the evidence is growing from multiple lines that's true. I also think it applies to iron 2 and clays, although that we haven't tested as, we haven't tested as many clays, and that these RMIs might be important. We can wrap up and just with some of the benefits on the last slide. I think ANA, I think it's applicable across a lot of contaminants of concerns. I don't think this is relevant only to chlorinated solvents. Many of the other things we're working with, even I have a student working with nitrite, Thomas Robinson. He's also been involved with this work. We're seeing a similar type of pattern there for chemodenitrifications. And the goal is, can we use some of these things? Can we use the redox potentials, the pH, iron 2 concentration, some of this SI analysis to be able to do that? And thank you. I'd be happy to take any questions.
Fantastic. Thank you, Michelle. We have received a number of questions, and I encourage our audience to continue to submit those in the Q&A box. First, Michelle, can the amount of ferrous hydroxide formed be correlated with its reactivity? So the more mineral present, is it more reactive? And And if that's the case, then would a goal be to increase the amount of ferrous hydroxide formation then?
Yeah, that's a good question. And at one point we tried to play with actually estimating in our bottles, right? Because we knew how much iron 2 disappeared and estimating how much, but then, you know, it really sort of drove us to two things. To one, that this notion that Paul brought up, that it's a dynamic situation here, right? So it's forming, but then it's rapidly being oxidized, right? It's rapidly reacting with something. So it's a moving target to try to get the mass of the ferrous hydroxide. I think Paul put it well when he said you rather need to sustain the conditions to continually generate the ferrous hydroxide. And I will add that we did do experiments, you know, because it does beg the question, is ferrous hydroxide itself the reductant, and like you don't need the magnetite, right? So if we're precipitating ferrous hydroxide on top of magnetite, like, all right, we'll get rid of the magnetite. And we did do, and that's in the paper in much more detail, we did the same exact solubility diagram in the absence of magnetite. And in the absence of magnetite, you don't see reactivity until you're very, very high concentrations of aqueous iron 2. You don't see them at the lower. So there is some sort of synergistic, or, you know, the magnetite is doing something, even if it's just contributing a surface to help it. But a good question.
Great, thank you. You showed that the active precipitation of the minerals is crucial. Because that's the case, would measuring the microbiology responsible for the active precipitation be helpful in getting information about ANA?
Yes. That I absolutely think. In fact, the title of this project, this ER2532, was, you know, we have to come up with acronyms, right? So the title of it, what was it? It was BMAD, biologically mediated degradation, whatever, right? So it's biologically mediated. Yeah, I think you can't. We're gonna have to look for the microbes, because they're going to be the ones likely generating the iron 2. And if you're looking at sulfide stuff, generating the sulfide. So yeah, I think you're going to have to be able to incorporate the presence of microbes that can help continuously generate the things that are needed to form these RMIs.
The next question is about the data you showed with the improved rates at high ferrous iron and at pH 8. Given that dechlorination can be an acidifying process, would the pH of groundwater perhaps need to be adjusted? Was it adjusted in your experiments up to pH 8?
Right, that's a good question. Yeah, so we did our experiments in buffer, right? And it's sort of a necessary evil, because if the pH flies around, then we can't really, you know, we can't nail things down. And so I think in the, you know, in the natural, obviously carbonate is serving as a buffer. I mean, I think the relative amounts of acidification that you would happen, I mean, I don't think those sites, you know, fluctuate heavily in pH and driving it down. You know, to me, the idea of actually controlling the pH in an aquifer, you know, adjusting it is a hard thing to do. But absolutely, if, you know, you have some levers to play with, right? And one of the levers is pH. And like we're taught, you know, in all our introduction to environmental chemistry kind of classes, like pH is indeed the master variable, right? It affects EH, it affects solubility of everything. And so I think, you know, being able to adjust pH is going to be one of those levers we have to look at.
Thank you, Michelle. You showed a graph a couple of times in your presentations. It was most recently on slide 60 that showed a really big jump in the rate constant at hitting the 0 mark. What caused such a large variation in the observed rate constant at that point?
Yeah, that's a good question. That's an astute observation, right? And so the the question is basically, so right along that zero here, right, whoops. Right along that zero here, you can see a wide variation in the rate coefficients there. And I don't know the answer to that question, right? So I think I think it's a case where you're right on the edge there. So that happened right at the, you know, on that line of that solubility plot right there. And so I think it's sort of like a tipping point. And, you know, you've either tipped a little bit one way or tipped a little bit the other way. You know, maybe the iron 2 concentration was measured just slightly off, or the pH was slightly off. And that sort of led to that. I think there's interesting things to be learned from that, but I'm not 100% sure. I don't know what they are yet, but an astute observation.
Sure. Understood. Thank you. The next question is that it seems like the magnetite and ferrous iron in clays does not reduce PCE and TCE, but what about dichloroethanes?
Yeah, that's the question. That to me is the question. And so, and we are working on that right now. When we've looked at the literature carefully on it, and I think there is a much more compelling case in just the literature data, nothing that we've done, that suggests that the DCEs could be reduced by magnetite. There's sort of, you know, there's the ferry work, which, you know, that's pretty compelling, that autoclave data that they have in there, and then they have magnetite alone data. There's some others that don't see it. So I think right now that's a mixed bag, and I don't know the answer, but we're looking into that, because I think that could explain some of the places where, you know, natural attenuation has been attributed to magnetite, where it might be that, you know, the primary mineral itself might be supporting ANA.
Got it. We have 2 more questions for you, and then we'll bring Paul back. First, when you talked about the addition of ferrous iron to the clay minerals, can you talk about how much ferrous iron was required, if you have that measurement?
Right. Yeah. So it was a lot. It was, I believe, Anka and Jim pushed it up to 20 milligrams per liter. So high iron 2 concentrations. And the clay was really interesting, because we found that, Anka found that when, if the clay itself had a high iron content, so one of the clays they worked with had 20% iron structurally, right? And we know from previous work that when iron 2 absorbs onto these high iron containing clays, electron transfer happens, right? And we actually, so the iron 2 that absorbs onto the clay gets oxidized. So then, you know, it's not going to be able to reduce PCE or TCE if it's been oxidized. And so, and she found that acts as a sink. So, right, so you have your aqueous iron 2, absorbs onto the clay, and then the clay with high iron sucks up those electrons, and she didn't see as much reduction of PCE and TCE. And it was in the lower clay, low iron containing clay, where there wasn't that sink for the electrons to go where she saw it. And then she did a very interesting experiment where she did it with a clay without any iron in it. So she just absorbed iron onto the clay with no iron present, right? So then no electron transfer, no sink for electrons to go in, and she saw quite fast reduction kinetics. So yeah. So high aqueous iron 2 concentration, but the complicated answer is, I think it's going to depend upon the iron. Her results suggest it's going to depend upon the iron content of the clays.
Great, thank you. It's widely known that the field measurement of redox potential, or ORP, is unreliable. Does that present a challenge to measuring the potential for RMIs in the field?
Yes, but I think people are doing very good work to overcome that. So yeah, ORP is, you know, just sort of a terribly flawed measurement in many ways, you know, theoretically as well as practically. And but this notion of adding these electron shuttles, you know, pulling samples and then adding the electron shuttles, I think that's showing significant promise. I think there's a lot of work going on different ways to measure redox potential that are going to fit into this new paradigm that comes out of this.
Thank you very much, Michelle, for the presentation and for answering these specific questions. Next, we will bring Paul back on the line and ask both of our speakers today a few general questions. The first question is about whether the focus on iron in this presentation is because the iron is unique for these types of reactions, or is it of interest due to its prevalence? And are there other systems with potential RMIs that haven't been studied as much yet? So we'll start with Paul, please.
The main reason that we're focused on iron is because it's ubiquitous, and so that's most of the answer to the question. And it's probably the most effective also. Is there any other alternative? That maybe is the question. Manganese? Not so much in this context. I don't know, Michelle has something to add to that?
No, not really. I mean, I think, yeah, in terms of minerals, I think here we kind of focused on, you know, iron oxides, or at least I did, and iron and clays, certainly iron sulfides, but I think iron it's the prevalent, and it's certainly the most redox reactive with some of these things. The other ones are either not prevalent enough or not particularly reactive.
Okay, thank you. And we had a request for a clarification. Are these results relevant only to anoxic aquifer conditions when oxygen is not present? Michelle, would you address that question, please?
Certainly the results I presented are only relevant to anoxic conditions. And I almost tagged that on to the end of the last question. You know, there is some evidence starting to come out of oxidative pathways in the presence of these iron minerals and clays as well. I think that's still, you know, very new and emerging, and we're not quite sure of that yet. But all of the results I discussed were certainly, and most of the framework that Paul was discussing, except in the beginning, I think is reductive in anoxic conditions.
Great, thank you. Paul, do you concur?
Yeah, actually, I just want to reinforce the idea that in a general sense, the idea of reactive mineral intermediates is highly applicable to systems that are not purely anoxic, and then, for example, in particular, this idea of active to passive transitions in an in situ remediation scenario, that's like an ideal situation to generate these dynamic formation and consumption of different mineral phases, and those are likely to be the most reactive phases, and oxygen may start to come into the picture at some point, and so those oxidative Fenton-like pathways might start to show up. We haven't got to that yet, so yes, it's out of scope for the specific details that we presented today, but it's on the horizon.
Okay, great, interesting. Michelle, we'll start with you for this question and then go to Paul. Would you please summarize what specific measurements you would encourage practitioners to make on field samples to better support E&E evaluations?
Yeah, right, that's the question. And that's the recipe I think we don't have yet. But given what we know now, I think there's a couple things I would look for. You know, I would look if iron is present, both in the soil and in the water, right? And so I wouldn't necessarily care if it's magnetite or, you know, iron oxides or something, but I would look if those were present, because I think you're going to need a source of iron into the water. You know, pH and EH. ORP. I would still make those measurements and see, you know, do you have, are you above the SI of some of these things that we would anticipate? And, you know, and then the microbes. I would look if there is something sustaining there. And then I think that information has to be then put into context of, okay, so we take those things and measure them, and then how do we decide? And for me, it would be, I would look for the presence of iron, and a source of creating iron 2+ in solution. And then I would absolutely, and we didn't talk much about this today, you know, iron sulfides are very reactive. In lab, what I see is the iron sulfides, things are very fast, and lots of the literature supports that. And there's the bird work that shows, you know, when you precipitate those. So, you know, the presence of sulfate and sulfate reducers would be another key thing. So I'm not sure it's anything new that you wouldn't have thought to look for before. But I think maybe look for it and then evaluate it in a bit of a different context.
Great. And Paul, anything to add to that?
Yeah, I just want to revisit the idea of the mediated ORP measurements. So remember that figure 28 that had the opposite slopes, and we should make it clear that in both cases the redox potential ORP measurement that's on the x-axis was measured in the presence of these electron shuttle mediator compounds. So now the issue for the person who asked the question is that going to go? Can that be translated to the field? And we hadn't done it, but I would point out that SERDP funded a project on this just the last round, and so I think the lead PI is Demin Fan, and Chris Coker is working with him. It's kind of a continuation of work coming out of my lab, but I'm a minor player in the project, and I don't remember the project number off the top of my head. But the whole point of that project is try to figure out whether they can come up with a protocol for doing these mediated ORP measurements in the field on environmental samples. And if they can do that, then that provides a real practical opening for trying to close the loop on all this.
Great, thank you both. And then the final question for today, would you please summarize the next steps in this research on abiotic degradation of chlorinated solvents? What are the most important next steps that you see? Paul, we will start with you, please.
Well, I guess I'll just use that to make an advertisement for the new project that we're starting, where I'm PI and Michelle is a co-PI, and in many ways is the integration of all this stuff that we just presented and the continuation of that. The two themes that are in that project that are new relative to what we presented today is the idea of a conceptual site model that integrates all this biogeochemistry. And so we're going to try to put the speciation modeling together in such a way that you could do the kinds of exploratory calculations that Michelle was describing a minute ago on specific field sites, even as a function of reactive transport. The other thing that's in that project was not in our scope until I just briefly mentioned the acronym a moment ago was active to passive transitions. And as we were writing the proposal for that particular project, we got really excited about the idea that this active to passive transition scenario, which is clearly of interest to SERDP now, is a super interesting and promising way to look for these RMI effects. And so we think those two things are going to go hand in hand in a way that will be quite useful and exciting and interesting.
Excellent. And Michelle, anything to add to that, or any other message you would like to leave our audience with today?
Well, I think that's a great ending. You know, I think our job now is to tell you what to do with these measurements, right? So we have to develop some sort of conceptual site model or some decision tree or some way for you to be able to go in and then say, okay, this site is amenable to ANA, or yes, it's okay to turn the pump off and transition to passive, because this will happen.
Fantastic. Thank you both. Again, Paul and Michelle, for the very informative presentations today and answering so many questions.
You're welcome.
Thanks.
Our next webinar, great, thank you. The next webinar in this series is on Thursday, December 10th, and it features DoD-funded research on efforts to measure and manage contaminated sediments. The webinar will feature Dr. Mandy Michelson of the U.S. Army Engineer Research Development Center and Mr. Gunther Rosen of the Naval Information Warfare Center Pacific. Registration is open, so please visit the SERDP and ESTCP webinar webpage to register for this and other webinars. Before we conclude, I would like to remind you that both the audio and a copy of the presentation of today's session will be archived on the webinar webpage in case you would like to refer to them in the future. I would appreciate it if you could please take a moment to complete the survey that will pop up on your screen at this time. This concludes today's webcast. Thank you.